With your permission, Mr. Baysal, I want to say information about you. He is working as professor of molecular microbiology at Muğla University in Turkey. He has carried out studies related to induction of resistance mechanisms of organisms using physiological and molecular parameters. His current studies are on host microbe interactions, molecular markers, biological control of pathogens, enhancing enzymatic properties of biogens using bioinformatics database, gene tracking and resistance to antibiotics. He is also interested in bioinformatic tools, drug discovery related to microbiology and molecular genetics. Professor Baysal is here. Speech title, How do we use bioinformatics for drug discovery to SARS-CoV-2? You can make your speech, Professor Baysal.
Okay, thank you very much for this opportunity. I would like to especially, first of all, thank to Professor Murat Gök and all the organization committee that has given to me a chance in order to give this talk and to share my findings with the valuable participants. I'm going to give you to talk about my current studies on SARS-CoV-2. As you know, in this pandemic situation, all the researchers are trying to find some solutions to stop this pandemic threatening to all the world. That's why I have already started some studies in the frame of my own facility. That's why now I would like to first give you some information about the bioinformatics, how we are using it in this kind of studies. And afterwards, I'm going to give you also some new studies and new findings that maybe some participants also can interested in these kind of studies as well. So my content of the talk will be first, what is the computational biology and how we're using the bioinformatics and what is the importance of data mining in bioinformatics database. And at the last of the talk, part of the talk, I'm going to give you the new finding on SARS-CoV. As you know, computational biology is the application of mathematical methods and computer algorithms to biological problems. And it can be also regarded as the branch of computational biology and bioinformatics. You know, bioinformatics is the main branch of this area. That's why we can also define it as the application of tools of computational systems, which can also be used for analyzing and interpretation of the biological data. We know that the biological data is very complex, and sometimes even we have lots of different available data, we cannot evaluate them, and we cannot also classify easily, because we need, at the same time, we need also mathematics, physics, and biology. All these scientific areas are also involving in this area, and it's a new science, you know. And it's at the interface of the molecular biology and computer science are coming together, and we need also to explore and analyze and understand these huge data. Is a world of genomic data. So bioinformatics seems to be a meeting point, you know, is also involving the computer science, mathematics, statistics, and biology and medicine. That's why that it has a very important place. But development in the bioinformatics as a methodology, we should also find the right milestones. I mean, the right way to understand how much we are also how much we continue with the right schematic following chart. So that's why we should also find, especially first, random model, and the run model, I mean, the running model should also be a property that should also carry some property as a validated quality. And after then, we can also continue our studies with refining model, and we should also provide it some strong data, I mean strange data, to prove our phytos and modeling. And then we're gonna also we should also continue with the formulated of hypothesis. Then, according to formulated hypothesis, we should also improve. I mean, we should also develop a new model that should be repeated again and to check whether run analysis is the correct or not. So, bioinformatics and data mining is also like partners, co-author partners, and the designing of the new algorithm and statistics for the large molecular biology data and set analysis. Informatics is a very useful tool, and the analysis and interpretation of the biological data, which also includes the nucleotide and amino acid sequence and protein structures, are also available, and also sampling for software tools. Without using software tools, we cannot combining, we cannot clustering, we cannot also observing what is the difference and what is the similarity and what is the common point of all these items. That's why bioinformatics, I think, is the mining of biological data sets. And bioinformatics is also a good tool for mining of the biological data sets. Why bioinformatics is necessary for biological science? For instance, we are dealing with the biological organisms, micro or microorganisms, but we should also find the exact data, compiling the data and evaluating of them correctly with the high quality parameters, which is necessary for understanding the real cases. It provides also us very good aspect to better understanding of the complex disorders. It can also be useful to solve existing of biological questions, waiting the answer. That's why we should also find the new way, and also we should also use the right algorithm and with the right software tools. And that's why we should also learn, how we can do better data management and visualization. And these are all dependent on experience that you can also get with your biological approaches. But not only biological approaches, but also the methods which you have already used is also providing very different aspects to evaluate them. So, as we know that the biological organisms very complex, macro or micro scale, but the most important thing, how we can also evaluate of huge data. When you check the life cycle of all microorganisms, microorganisms or macroorganisms, they have always providing, supplying the different behavioral rhythmic differences that depend on their environmental condition, adaptation, etc. But the most important thing which affecting all organisms is environment. And our environment has an important effect on the smallest molecule, the DNA, and DNA contains the huge data. That's why you should. The DNA is the raw data, is not being transferred, is not being framed. That's why the second step of the DNA will continue with continues with the transcription, and transcription can be followed by the gene expression, and the transcription. One step further, we can, one step after transcription is the translation. And translation means the protein formation and protein structure. So protein structure can be followed with the proteomics. And after the protein determination, we can also continue our study with the biochemical cycles, and following of the biochemical cycles can be available by data obtained by the metabolomics. And all these kind of different circular systems involving transcription, translation, proteins, and biochemical cycles. At the end of the all organism actions will be ended with the phenotypic formation that depends on inherency. I mean the genetic locus differentiation, locus transferation, vertical or horizontal gene transferring. All these things all these things are forming the last form of the biological structure. So as you know that these huge data, from the one side, you can get lots of information in DNA, you can get lots of information for the transcription, translation, and metabolomics, proteomics, and gene expression. I mean, now in this century, we are also providing lots of huge data related to whole genome expression, which can also be described as a descriptive data. That means the RNA seq. And the huge data cannot be compared with each other without any help of computational databases and computational evaluation as well. So that's why gene expression, proteomics, and metabolomics should be combined together, but by using of the right tools. Types of data available now in this century when the molecular biology, genetics, and the bioinformatics are also involved in this kind of activity based on DNA, RNA sequence, protein structure, protein function, etc., and disease information. You know, now in this century, lots of different disease information can be related easily with the gene expression and molecular pathways and the proteins which are responsible for activation of some disorders, etc. So that's why these kind of huge data should be combined with each other, and we need also the exact tools to combine them. On the other hand, you know, in this century, as well as the computational tools, the huge data is yielding and accumulating somewhere, and it also grows very quickly day by day, because all the world are also trying to find some solutions to solve these kind of problems, but they're also sending their findings to do a spatial storage. That's why it also causes the large peak of data growth. And popular databases and type of data where the old data stored is mainly on GeneBank, UniProt, Protein Data Bank, Gene Ontology, DCSSP, and CAC pathways. These are all also storing the data where all these kind of biological process are following. And you know, but on the other end, we are also leaving some problem while we're doing some analysis. And the problem affecting the analysis, sometimes they direct us to do some wrong direction. That's why we should also be sure of data quality, and database quality is of importance to correct interpretation, using the correct parameter, spelling. I mean, avoiding of the spelling mistakes, and to avoid of annotation errors, and to understand where the frame shift errors are occurring, etc. That's why we cannot be 100% sure that when we use any kind of analysis, we cannot be sure that whether it is correct or not correct. So the problems, one of the big problems is also what we are facing with is the database redundancy. Everybody is, I mean, high-level researcher in their labs or working day by day, and they are also collecting the lots of different data, and they are sending to do some data sources their data, what they have already got it, but sometimes the redundancy, you know, is necessary. And there are lots of problem because of the redundancy. Sometimes also you can also download some data and you cannot be sure whether it's 100% correct for your studies. That's why we must also strain it. I mean, we must screen the right data which is available and which is the correct data for our further studies. That reason, As a result of redundancy, if the data used for training and testing are closely related, we must check it. Otherwise, it can also be directed to us very different points that are not related to our studies. And the developed model will lack ability to generalize. That means that you are fail. You cannot reach the exact point where you want to be. So that's why you must be careful. And now when you check the future of omics science, the omics science at the beginning of the 19th started with the genomic studies. All the sequence data which are available from the some microorganisms, there was a very big, huge data accumulation because of the genomics, and it also continues up to now. But at the round of after the 1905 and between the 2000, a new study has started to accumulate cause of the proteomics, because now we have already understand the whole protein expression of each organisms and how the protein are changing depending on abiotic and biotic stress subjected to any kind of the biological stress, which is also exposed on any kind of microorganisms. And abiotic and biotic stress is the reason of protein expression differentiation. So the proteomics should also be followed. When we started to look at the whole genome sequencing, it can also be not provide the exact point, because we cannot be sure how many % these nucleotides are also translated to the protein level and protein formation. That's why we must be sure. For instance, in the eukaryotic cells, we are living different phases between the transcription level and translation level, called as post-translational modification. Each protein formation are also different, and depending on this modification process, changing their property. At the end of the proteomics studies, genomics and proteomics may be combined together, and they are providing very good tool for us. But the most important thing is the system biology, and to combine them and to check how many % we can get the right data from the proteomics, how many % we can get the right data from genomics. That's why system biology should bring them, all of them together, and to show us what is the right answer for our questions. And even perspective in biology has been started with the structural biology. Then the studies has converted to another dimension related to modeling. Now the most important thing, what we have already modeled, should be also available for us, should be also beneficial for us to estimate of some cases. Even we don't know exactly, but it should also give us a high probability accuracy for understanding of some cases that can be described as a prediction. Now the prediction is a higher value for us for understanding of any kind of biological processes. We have already now structural data, modeling data. We modeled everything, and we know how, at least we can use some model organisms to use them as a model organism. But the most important thing, what will be our estimated findings when we use this model on a kind of different organism which has not been tested before. This is most important. So bioinformatics at the beginning of the 19th was the style with this classic. Then it continues with the biosimulation. And now in this century, we should also find the predictional methods, how we can use the biosimulation and classical ways. That's why bioinformatics is developing day by day, and the prediction is much important. Is becoming much important than before. So, you know, why that prediction is important, because data interpretation is a comprehensible cases that involving the genome sequence, mRNA expression, and the 3D structural predictional databases related to protein, and the crystallography and electron crystallography is developing very quickly. And when we got some information from the mass spectro, considering the 2D dimension, we can understand from the tag sequence of each protein and how these kind of proteins are also playing a role to establishing of the last conformative data of the protein. So all these things are very useful and beneficial information for understanding of any kind of cell, such as bacteria cell, eukaryotic cell, and animal high organisms. So there are lots of different complex data involving the biological pathways, and some of the DNA description, some of the mRNA description, some of the protein description are very different. So if we are able to understand what is going on, then we can find lots of different solutions easily. What is threatening to us, right? For what is threatening to us. So genomic data in evolution. After getting some information about the transcriptional phase, gene expression phase, and proteomics databases, the second phase of these kind of studies related to molecular docking. You know, when we get the right 3D protein structure, when we got the right data of the genomic databases, when we get when we also try to understand how many percent we can use this gene product to do this problem, that's why we need molecular docking. And searching of the algorithm and systemic approach, especially the matching percentages of each kind of protein ligand interaction based on the genetic algorithm, will provide us how many percent we can use these data for cure of any kind of problem, cure of any kind of disorders, what is our problem is. So force field, empirical and knowledge bases, these should be also combined with each other to understand the docking assessment. And the right docking accuracy will provide us how many percent we can use this approach to designing of the right designing drug candidates. So computational drug designing approach has a different aspect, but the most important thing is the use of the right screening method, protein docking analysis, genomic data. When you know the target pathogen, when you know the target cases, which is also be waiting for your solution, is the characteristic property in available genome data will be your most important contributor, most important assistance for solving of this kind of problem. When you find the right characteristic data and available genome data, you can also combine with them using of the PDB databases, the right protein selection, and so on. Then you can get also very good, the computational drug designing approach could be also expanded by the study with the studies on in vitro assays, and these in vitro assays should also be transferred to do in vivo assays, because even we got the very good results in vitro assays, it doesn't mean that we can find the right responses, what we have already got it in the in vitro assays, and we cannot be sure how many percent we can also find the right analysis results on the right host. that's why we should also approve the all analysis in vivo assays. So the steps related to docking and virtual docking and homology modeling, and depending on the potential drug candidates, I mean, determination of the potential drug designing and drug candidates, there are different databases are available, and different methods based on biological calculation techniques are mainly presented in, and they are already developed. And day by day, it's also going to get the high level for prediction of these kind of problems and for these kind of evaluation. The most important thing, what is the now mainly interested by all the researchers before the in vitro assays to find the exact compound, which which shows the interaction with each other, and it based on target and ligand, and the protein and ligand interaction, and protein-protein ligand interaction. So what we have done on SARS-CoV-2, I have already explained all of these tools related to bioinformatics and how we can use it and so on. As you know, that the study on SARS-CoV-2 is not easy because it's very dangerous and it's very contagious pathogen, And to work on this virus, you need some special equipment and special laboratory conditions, which can be described as a security tree. So that's why you cannot work easily without any permission, without any getting the right conditions to work on it. That's why in this pandemic period, while the pandemic started from the China, especially from the Wuhan, I have already thought that what can I do by myself? Even I have no facility, I should also do something, Even I have no facility to work on the virus easily because I have no facility to work it. I have more or less I have some knowledge on bioinformatics, and then I have decided what can I do by myself. And I think bioinformatics is the good tool, even you have no facility to work on it in vivo conditions. Then I have decided to collecting of the full genome sequence data of the SARS-CoV-2 of the databases belongs to Germany, GSID, and I have downloaded all the gene sequences. That's very funny, but it's the real. There are more than 614 different whole genome sequence of SARS-CoV-2 was available, and we downloaded. But the most important thing is the working with the right NGS data. You can download lots of different data. When you cluster it, you find nothing because the genetic sequence quality is very low and shows very big lacks, very big lack of some sequences. It shows some fails, not very useful data. That's why we screened all the data and we find the exact full genome data describing the properties of the full genome of SARS-CoV-2. Afterwards, we did some clustering, and then we continue our study by the mutation screening. We use the phylogenetic trees, and as you know, all those studies, what we have already understand from the clustering, and which is aim for the clustering analysis. Most of the studies are done with the cluster analysis. But when we check the cluster, when we check the sequence by the cluster analysis, we didn't find the very big important data that can be also classified as a common gene regions, And then we decided to test the another analysis method called MAO. Then we used the MAO. We have found that the clustering is totally different. Afterwards, when we get the clustering databases considering the MAO analysis, we have subjected to all these kind of sequence as a pairwise alignment, considering the global needle-man wound. And we have found some stable regions here, as you have seen, from the beginning of the sequences until the 600, 500 data bus pair long, all the sequences showed some stability. And it doesn't change. It doesn't change. And then we said, something must be here. I don't know what's going on, how it can be possible. Then we try to understand what is the main reason for this stability of the sequences. After we have determined what is the stability of sequence and what kind of genes are also present here and what they are coding. When we check, we find the spike protein. I mean, we check when we found a protein sequence encoding the sequence of a spike protein. But this is very interesting, you know, when we give the analysis of the data obtained from the SARS-CoV-2, the database has suggested us that this data belongs to feline infection perinatus virus, and this virus belongs to, I mean, is the viruses of the cats, not the humans. But that is very interesting. It is also a spike protein. I mean, this code is also encoding the spike protein. I mean, when we check it, and then we submitted to protein databases and considering the EITizer databases, which is a very big server, and also using the artificial intelligence, it shows that these protein data has a big similarity with the 6G6X7A. And then when we check, these proteins has a very strong interaction, has a very strong computational competency with the N-acetyl-d glucosamine. And then we have already thought if this protein, which has already submitted from SARS-CoV to data server, and data server has given us that our protein sequence belongs to feline perinatus virus, but even it shows that it's a protein sequence of the feline perinatus virus. It has also competency as a protein ligand, and this protein ligand is N-acetyl-d glucosamine. And then we have already thought that maybe it can also be a good solution for us. Why we cannot use the for SARS-CoV-2? Why we cannot use these ligand? Then after we checked whether the possibility is, how many percent possibility is present for us. Then when we check the ORF1A region in SARS-CoV genome, this stability region, what we have already found it, is very unique. You know why? As you have seen in the presentation, when you describe any kind of genome which has a relationship with the SARS-CoV and other viruses, if you want to diversify, if you also want to separate them, you can see here the SARS-CoV-2, MERS-CoV, and 2000N and CoV can be described after this ORF1A or RF1B or F1A, and it starts here. Before this sequence, you cannot be described it as SARS-CoV, MERS-CoV, or 2000N, because the ORF1A and ORF1B is stable in all viruses. Then we said, oh, we are in the right way. Why not? It's possible. Then we found that this genetic stability is very interesting. And if we can use this genetic stability as a target, where our component can also attach to this side of the genome, it is also possible. It may be possible to stop the viruses. At least we can also break up the compatibility with the ACE2 receptor and the virus integration. We can break up. So that's why then we decided how we can also, this coronavirus enters the cell through the ACE2, this receptor. If we can load the coronavirus spike protein with the N-acetyl-d glucosamine, it could be possible to stop the viruses, and it could be also inhibited for integration into the cell. Then we check. We have seen that a number of ligands, when you load it on the virus protein, it's accumulating on the virus protein, and the most important proteins playing the role in pathogenicity of the SARS-CoV, N-acetylglucosamine is the right ligand that can also be attached on the virus protein. And afterwards, we continued our study with the different databases based on the antibody. As you know, normally we are producing our antibody in our system, and against some microorganisms invading to our body. We are always facing with the microorganisms every hours in 24 hours in a day. But the most important thing, how many % we can get the protection against the invasive microorganisms. We know there is no higher possibility to increase the antibody amount of the body against the viruses. But we should also find a way how we can introduce our antibody that they can easily find the virus easily in our body when it enters to our organism. Then we have modeled our system based on antibody. We have seen that the antibody, when the virus loaded with the N-acetyl-d glucosamine, it provides that antibody finds the virus much easier and make a contact, make an interaction very easily than the previous phases. It means N-acetylglucosamine, depending on its increasing of its concentration in this circular cases, it provides that N-acetylglucosamine shows the antibody where the virus is, and it shows also it also shows the antibody where the virus is and how it can also bind to virus. We have already published our finding in 2 articles, but I would like to especially imply and stress that the first publishing was not easy, because all the researchers studying on these viruses has also claimed that it is not possible. No, it is not possible. It is not possible, and they have rejected very quickly without reading my article at the first phase of my submission. But I didn't give up trying, and I have found I have already tried to journals, and I have write many different explanation letters to the different reviewers of the journals. At the end of the I'm not at the end of, but after trying to find the right publication, the Germany papers, journal belongs to the Germany, has accepted my first article. And then we achieved to publish it. When I publish it, then we continue our study. We didn't give up, and we continued our study on the antibody recognition. And in this level, we achieved to publish another article that, in this case, we were able to show that the antibody, when it combined with the N-acetyl-D glucosamine, when the virus loaded with the N-acetyl glucosamine, the antibodies of the cells are finding the virus very easily, and we proved. Then we also publish it. So then another paper which is also available, if someone and any researcher, any young researcher interested in, can also read it. This is our last chapter, which I have already written. And these are all my studies and activities during this pandemic period. And I have wanted to share it with all if someone may be interested in, I don't know. So thank you very much for listening. And I would like to special thank for the opportunity that